Welcome to the Deep Dive. Today, we're heading into the microscopic world, specifically looking at Salmonella Enteritidis in poultry from Argentina. We've got a really interesting piece of research as our source, Hoffman et al. 2025.pdf, which was published in the International Journal Veterinary Microbiology.
Exactly. And, this deep dive is really all about getting to the heart of this study. We're focusing on what it tells us about the genetic makeup and, crucially, the antibiotic resistance of Salmonella Enteritidis strains found on Argentine poultry farms. Our goal is basically to understand the current situation with this particular bug, in that part of the world.
And this is a big deal because, well, as you know, antibiotic resistance is a major global health threat.
Yeah.
Especially when it comes to bacteria that can move from animals to us through the food chain. Salmonella enteritidis, or SE, is a prime example, causes a lot of foodborne illness worldwide.
Absolutely.
So understanding what's happening with it in a major poultry producer like Argentina is something we really need to dig into. Okay, let's start with the basics. Salmonella enteritidis and why we're so concerned about it in poultry.
Well, Salmonella enteritidis is indeed a leading cause of food poisoning around the globe. What's particularly interesting, maybe, is that in chickens, the infection often doesn't show any outward signs.
Really?
Yeah, the birds can be carrying it without appearing sick at all, but they can still contaminate eggs and meat, which then obviously creates a risk for us.
So it's like a silent threat in the poultry flock?
Pretty much.
And it's not just about human health, right? There must be a significant economic impact for the poultry and egg industries when contamination or outbreaks happen.
Oh, absolutely. Beyond the immediate costs of dealing with contamination, there can be big effects on trade and, of course, a hit to consumer trust.
Yeah.
The study also touches on the idea of how SE might have spread globally, potentially through the international movement of breeding animals and the centralized way the industry often sources its stock.
That makes sense in our interconnected world. Now, Argentina itself, why is it such a key place to study this?
Well, Argentina is a major player on the world stage when it comes to poultry. It's the 8th largest producer and exporter of chicken meat.
Wow, 8th largest.
Yeah, and plus they have a very high rate of egg consumption per person. Another key factor is that most of Argentina's poultry industry, actually over 80%, is concentrated in just 2 provinces. Buenos Aires and Entre Rios.
80% in just 2 provinces.
And that kind of geographic concentration can really have an impact on how diseases spread.
Interesting. A lot of poultry production in a relatively confined area. The study mentions that SE has been linked to poultry production in Argentina before.
Yes.
And that it's even part of their national poultry health plan.
Yes. Previous research has definitely shown that SE is present in Argentine poultry. The fact that it's included in their national health plan tells you it's a recognized issue. What makes this new study by Hoffman and colleagues so important is that despite this awareness, this is actually the first time a study has really looked at the genetic makeup, the genomics, of SE strains isolated specifically from Argentine poultry farms.
So it's filling a crucial knowledge gap.
Exactly. A crucial gap.
Okay, so this research is groundbreaking in its focus on the genetics. Let's get into how they actually did this, the scope of the study and the methods they used.
Okay, so the researchers focused on 20 SE strains that were collected from poultry farms in Entre Rios province. This was between January 2020 and February 2021. They took samples from several areas within Entre Rios, including places like Colón, Gualeguaychú, Paraná, San Salvador, and Uruguay.
And where exactly on the farms did these samples come from? What kind of samples?
Right. The majority of the samples, actually 16 out of the 20, were from swabs of the litter in broiler houses.
Okay, the bedding.
Yeah, and interestingly, most of those came from just one single poultry company. The other samples included 1 from dust in an automated cage system for laying hens and 3 from laying hens themselves, specifically from their feces and cloacal swabs.
Got it.
And then they put these 20 isolates through some very thorough testing.
Tell us about that testing. What do they look for?
Well, they conducted what's called phenotypic testing for resistance against a really wide range of antibiotics, 50 different ones, actually.
50?
Yeah, covering 14 classes of antimicrobials that are important in both human and animal medicine. This involved using a standard method called disc diffusion on agar plates, and they interpreted the results using guidelines from CLSI, which is the Clinical and Laboratory Standards Institute, and also ANLIS, the Argentinian equivalent.
Okay.
For one particular antibiotic, colistin, they use a slightly different method called a microbroad disc elution test because the standard disc method wasn't really established for colistin back then.
50 antibiotics.
Yeah.
That's a really comprehensive screen. And then they went even further looking at the bacteria's DNA, right? They used whole genome sequencing.
Exactly. After they saw how the bacteria behaved in the presence of antibiotics, that's the phenotypic part, they used whole genome sequencing, or WGS, to analyze the complete genetic code of these strains.
Okay.
This allowed them to look for the specific genes responsible for any resistance they observed, and also to see how closely related the different Salmonella strains were to each other. This DNA data was then analyzed using some powerful computer tools, some bioinformatics.
Okay, let's break down that DNA analysis a bit. What did that involve?
Well, they compared the DNA sequences of their 20 SE strains to a huge number of other Salmonella sequences that are stored in big international databases like Enterobase and NCBI.
Right, these huge genetic libraries.
Exactly. Think of them like massive libraries of bacterial DNA. The researchers were specifically looking for changes or mutations in genes called NFSA and NFSB, which are known to play a role in resistance to an antibiotic called nitrofurantoin. They used a software program called Genius Prime to help with this detailed comparison.
Nitrofurantoin. Okay, we should definitely remember that one. What else did they do with all that genetic information?
They also looked for plasmids. Now, plasmids are like these little extra loops of DNA that bacteria can have separate from their main chromosome.
Right, mobile elements.
Exactly. And what's important about plasmids is that they can often carry genes for antibiotic resistance and can even be passed between bacteria.
Which is how resistance spreads so quickly sometimes.
Precisely. The researchers used a tool called Platon to predict which bits of the DNA might be plasmids. Then they assembled those sequences and compared them to known plasmids in the NCBI database. They also used specialized tools called Plasmid Finder and Abricate to classify the plasmids into different families, and something called PMLST, which is a way to further subtype them. It gives them a very detailed picture of the mobile genetic elements present.
So a really in-depth genetic investigation looking at both the core DNA of the bacteria and these extra bits of DNA that can spread resistance. Now let's get to what they actually found. What did the phenotypic antibiotic resistance testing reveal first?
Okay, so the study uncovered some really interesting resistance patterns. On a positive note, all 20 strains were susceptible to several important antibiotics.
Good news first.
Yes, things like ceftazidime, avibactam, carbapenems, streptomycin, amikacin, azithromycin, chloramphenicol, tetracycline, and colistin. These are crucial for treating various infections, so it's definitely good news that they didn't see any resistance to these in their sample.
That is definitely reassuring, but I have a feeling there's a but coming, right?
You're right, there is. One of the most striking findings was that every single one of the 20 strains, it's 100%, was resistant to nitrofurantoin.
100%.
100%. And what's really striking about that is that nitrofurantoin hasn't been used in Argentine poultry since 1995.
It's 1995.
Yeah. It's a really stark reminder of how long antibiotic resistance can persist in bacterial populations, even after the selective pressure, the antibiotic itself, is removed.
Wow. 100% resistance to an antibiotic that hasn't been used for decades. That's a major takeaway right there. What else did they find in terms of resistance?
Well, they also saw high levels of resistance or reduced susceptibility to fluoroquinolones. A concerning 90% of the strains showed this.
90%, that's high.
Very high. Specifically with ciprofloxacin, 10% were fully resistant, and a large proportion, 75%, showed intermediate susceptibility. That means the antibiotic might not work as well.
Right, it might fail in treatment?
Exactly. They observed similar trends with other fluoroquinolones like afloxacin and enrofloxacin. And these are important drugs often used to treat more serious salmonella infections in humans, so these findings are definitely worrying.
Fluoroquinolones are a key class of antibiotics, so high levels of reduced effectiveness are a serious concern. What about other types of antibiotics?
They also found significant resistance to sulfonamides and nalidixic acid, with half of the strains, 50%, being resistant to both. For other antibiotic classes, the resistance rates were lower, but still present. 45% showed some level of non-susceptibility to tetracyclines and merlactams, 5% to phenols, and 5% to aminoglycosides.
So it's a mixed picture, really. Some important antibiotics still working, but significant resistance to others, particularly that nitrofurantoin and the fluoroquinolones. Did they see different combinations of resistance in the different strains?
Yes, they identified a total of 18 different patterns of antibiotic resistance across just those 20 strains.
18 patterns in 20 strains.
Yeah, quite diverse. The most common pattern, though, was resistance to both nitrofurantoins and quinolones. The number of antibiotics each strain was resistant to varied quite a bit too, ranging from just 2 up to a very concerning 19.
19. That sounds like multi-drug resistance. Did they look into that specifically?
They did. They found that a high percentage, 70% of the strains, met the definition of multidrug resistant, or MDR.
And MDR means resistant to?
It means being resistant to at least one antibiotic in 3 or more different classes or categories of antimicrobials.
Okay.
However, on the slightly better side, they didn't find any strains that were classified as extensively drug-resistant, or XDR. They did identify one strain called Argino-8 that was producing ESBLs.
ESBLs?
Yeah, these are enzymes, extended-spectrum beta-lactam that can make certain important bocaplactam antibiotics, like some cephalosporins, ineffective.
Okay, so we have a clear understanding of how these Salmonella strains are behaving in the lab when exposed to different antibiotics. Now let's connect that to the genetics. What did the genomic analysis reveal about this resistance and the plasmids they found?
Right. The genomic analysis helped them pinpoint the specific genes responsible for some of the resistance they observed phenotypically. For example, they found the ACKY gene, which can confer resistance to aminoglycoside antibiotics in all 20 strains.
In all of them?
In all 20. They also identified the BLAST-TTXM14 gene, which is a common ESBL gene responsible for resistance to ball-lactams, but only in that single ESBL-producing strain, Argino-8.
Okay, so that matches the phenotypic result for ESBLs. And what did the DNA tell them about the fluoroquinolone resistance?
For quinolone resistance, they found specific mutations in a gene called gyrase, which is part of the bacteria's main chromosome. They also found the QNRB19 gene, which is often located on plasmids, in some of the strains that showed that intermediate or full resistance to fluoroquinolones in the lab. And importantly, they also confirmed a specific mutation in the NFSA gene called PQ180 in all 20 strains. This provides a really clear genetic explanation for that universal resistance to nitrofurantoin that they observed.
So a direct link between the genetic code and the observed resistance for nitrofurantoin, even though the drug isn't used.
Exactly. The gene mutation persists.
Were there any other interesting genetic findings, anything unexpected?
Well, yes. All the strains also carried GALD-S and GOLD genes. These are associated with resistance to heavy metals like gold and copper.
Gold and copper.
Yeah. While it's not directly related to antibiotic resistance in the way we usually think about it, it's an interesting side finding. It might suggest these bacteria are exposed to these metals somehow in the poultry environment, maybe through feed or equipment, perhaps.
Interesting indeed. Now let's talk more about those plasmids, those extra bits of DNA that can spread resistance. What did the study find there? How many? What types?
Okay, so they detected a total of 10 different plasmids across the 20 Salmonella strains. 2 of these belong to a group called INC-F, and 1 belonged to the INCI-1 group. That INCI-1 plasmid, notably, was the one carrying the black TTXM14 ESBL gene we mentioned earlier.
Ah, okay.
Interestingly, they couldn't fully identify 2 other plasmids using the available genetic databases, but their DNA structure strongly suggested they were plasmids. And for 2 of the Salmonella strains, they couldn't actually determine what plasmids, if any, were present based on the sequencing data.
So a variety of these mobile genetic elements were present, some known, some perhaps new. Were any of them particularly common or carrying specific genes that are concerning?
Well, the PSEN plasmid, which belongs to that INC-F group, was the most common. It was found in 16 of the 20 isolates, so 80%.
Okay.
Now, this particular plasmid, PSEN, is known to carry several genes that help Salmonella cause disease virulence genes.
Ah, not resistance genes.
Primarily virulence genes in this case. Things like ARCIC, the PVT operon, and the SPVCBR operon. These genes are involved in things like helping the bacteria resist the host's immune system and being able to invade the host's cells.
I see it.
So in this specific instance, the most common plasmid seems to be more associated with the bacteria's ability to cause infection, its pathogenicity, rather than with antibiotic resistance directly.
So virulence seems to be a key feature of the most prevalent plasmid. What about that INCI-1 plasmid that carried the lactam resistance gene, the ESBL gene?
Right. That INCI-1 plasmid, specifically of a type called PMLST19, was found in that one ESBL-producing strain. And as we said, it carried both the AF3-HAMA gene, conferring some aminoglycoside resistance potential, and the BLAST-TTXM14 gene for lactam resistance. What's also significant is that this is apparently the first time this specific PMLST type, PMLST19, of an INCI-1 plasmid has been reported in Argentina.
First time reported there.
Yes. which really highlights how new resistance elements can emerge and be identified through this kind of detailed genetic analysis. It shows things are constantly changing.
That really underscores the dynamic nature of antibiotic resistance, doesn't it?
It really does.
What about the QNRB19 gene for quinolone resistance? Was that also located on a clearly defined plasmid?
Well, the QNRB19 gene was found on some DNA fragments or contigs that looked like plasmids in several isolates. However, the researchers couldn't determine their INC group or PMLST type using the standard databases.
So maybe on a novel plasmid.
It suggests that there might be new or less well-characterized plasmids circulating that are carrying this quinolone resistance gene. More investigation needed there, potentially.
This is getting into the real nitty-gritty of the bacterial genetics. So we've looked at the resistance patterns, the genes, the mobile genetic elements. Now, what about how these different Salmonella strains are related to each other genetically? Do they all look the same?
Well, at a broad level, yes. All 20 of the SE strains in this study belong to the same sequence type called ST11.
ST11, is that common?
Yes, ST11 is a really common type of Salmonella Enteritidis found pretty much globally. However, when they did an even more detailed analysis looking at tiny single-letter differences in their DNA, these are called single nucleotide polymorphisms, or SNPs, they found some interesting patterns.
Okay.
The strains tended to group together based on where they were isolated from. Specifically, there was a clear genetic separation between strains that came from broiler chickens and those that came from laying hens.
Broilers versus layers. They were genetically distinct.
They formed distinct clusters, yes. This suggests that there might be different populations, or maybe slightly different lineages, of Salmonella circulating in these two different sectors of the poultry industry.
That's fascinating. Can you tell us more about that clustering? Any geographic patterns?
Yeah, within the laying hen isolates, they even saw further subgroupings based on the specific county where they were found, particularly for those from the Paraná area. The broiler isolates, on the other hand, seem to be more widely distributed genetically across the different counties they sampled. They didn't cluster as tightly by location.
Huh. Any thoughts on why?
Well, it could indicate potential links to specific farming practices, maybe different management systems or environmental factors within the broiler and layer industries that might be favoring the selection or persistence of slightly different genetic variants of Salmonella in each sector. Or maybe how birds are moved around.
That makes a lot of sense. Different environments, different pressures could certainly shape the genetic makeup of the bacteria. Did they compare these Argentine strains to Salmonella from other parts of the world or even neighboring countries?
They did, and interestingly, they didn't find any close genetic matches between the strains in this study and SE genomes that were available from poultry, livestock, or wild animals in nearby South American countries like Chile, Paraguay, Uruguay, Brazil, and Bolivia.
So not closely related to their neighbors.
Not the available data, no. However, perhaps surprisingly, they did find significant genetic similarities, meaning very few DNA differences, like 12 SNPs or less to Salmonella strains that had been isolated from human infections, clinical cases in the USA and the United Kingdom.
Wow, that's a potential link across continents, from Argentine poultry to human cases in the US and UK?
Well, the study shows a close genetic relationship. It doesn't definitively prove the direction of transmission, of course.
Right, correlation, not causation.
Exactly. But the genetic closeness is striking. The study itself doesn't provide a definitive answer on how these geographically distant strains might be related, but it certainly raises the possibility of global connections and the spread of Salmonella Enteritidis.
How might that happen?
It could potentially be through international travel, the trade of food products, or even the movement of breeding animals like we mentioned earlier. The researchers mentioned specific identification numbers for those closely related strains, if anyone wants to look them up in the public databases. PNUS 333652 and PNUS 332477 from the USA, and PNUA 20011288849.1 from the UK. This finding really highlights the potential interconnectedness of the global spread of these bacteria.
That's a really key finding with potential implications for how we think about food safety on a global scale. Okay, let's move on to discussing the broader implications of all of these findings. What are the big takeaways here? What does this all mean?
Okay, well, one of the most significant implications is definitely that high level of resistance to nitrofurantoin.
The 100% resistance.
Yes. In all the isolates, even though it hasn't been used in Argentine poultry for nearly 3 decades, this resistance, as we discussed, is linked to a stable change in the NFSA gene on the bacterial chromosome, which isn't easily transferable to other bacteria via plasmids.
Right.
What this really shows us is how resistance to an antibiotic can become fixed and persist in a bacterial population long after the antibiotic itself is no longer used. It's a crucial point to remember when we think about managing antibiotic resistance. Stopping use doesn't mean resistance just disappears overnight.
That's a really important point. Resistance can be a very long-lasting legacy issue. What about the connection between the genes they found and the actual resistance they saw in the lab? Was it always a direct one-to-one match?
The study highlighted, I think, a complex relationship there between the genetic makeup, the genotype, and how the bacteria behaved when exposed to antibiotics, the phenotype.
Okay.
For some antibiotics, like nitrofurantoin, the presence of specific genetic changes directly explain the observed resistance. Very clear link. However, for other antibiotics, predicting resistance based solely on the presence of known resistance genes wasn't always straightforward. Meaning there might be other resistance mechanisms at play that we don't yet fully understand at the genetic level, or maybe the expression of those genes is regulated in complex ways.
So just because a bacterium has a gene that's known to cause resistance, it doesn't automatically mean it will be resistant in a lab test, or maybe not resistant to a high level.
Exactly. That's a great example they found. While all the strains had the AEP-AJA gene, which is associated with aminoglycoside resistance, only one strain actually showed phenotypic resistance to kanamycin and aminoglycoside in the lab.
Only 1 out of 20, even though they all had the gene.
Right. This illustrates that the presence of a resistance gene doesn't always translate perfectly to active resistance. There can be other factors involved, like gene expression levels or other genetic background effects.
What about the quinolone resistance with those gyrase mutations and the QNRB19 gene? How did that correlate with what they saw in the lab?
There, the correlation seemed a bit clearer. They observed different levels of resistance or intermediate susceptibility to quinolones in their strains, and these genetic factors, the chromosomal gyrase mutations and the plasmid-mediated QNRB19 gene, seem to be the main drivers.
Okay.
However, they also noted something interesting. In their study, there wasn't a clear link between resistance to ciprofloxacin and the production of ESBLs.
Really? Versus times those are linked, right? On the same plasmids.
Exactly. Some other studies have found such links, often because the genes are located on the same mobile element. The fact that they didn't see that strong correlation here suggests that the ways Salmonella develops and combines resistance mechanisms can vary in different bacterial populations and in different geographic locations. It's not always the same story everywhere.
And the lactam resistance, the ESBL stuff, seemed relatively low overall in the study, even with the identification of that one ESBL-producing strain.
Yeah.
What are the implications of that finding?
Yes, both the actual phenotypic resistance to lactam antibiotics and the presence of the BLAST KTXM14 gene were relatively low in this particular set of strains, found in only that one isolate.
So maybe not a huge problem yet in this population.
Perhaps not widespread in this specific sample. However, the researchers rightly emphasize that it's still really important to continue monitoring for these types of resistance genes, like ESBLs, because they are often on plasmids and have the potential to spread rapidly among bacteria once introduced. Low prevalence now doesn't guarantee low prevalence tomorrow.
Good point. Constant vigilance needed. What about the high prevalence of those INCEF plasmids, the PSCN ones, that were carrying genes associated with the bacteria's ability to cause disease, the virulence genes? What does that tell us?
Well, the fact that these plasmids were so common, found in 80% of the isolates, even if they weren't primarily carrying antibiotic resistance genes in this study, is still significant for food safety.
How so?
Because these virulence genes could potentially enhance the ability of these Salmonella strains to cause illness in people if they contaminate food. It highlights that we need to consider not just antibiotic resistance, but also the overall disease-causing potential, the pathogenicity of these bacteria circulating in the food chain.
Right. It's not just about whether we can treat the infection, but also how likely it is to cause infection in the first place.
Precisely. And the detection of that NCI1 plasmid carrying antibiotic resistance genes, the ESBL and aminoglycoside resistance genes, which has been linked to human cases in other South American countries. What's the importance of that finding?
That seems quite significant.
It is. That's a key finding that really underscores how antibiotic resistance can potentially move between different bacterial populations and, crucially, between animals and humans. The fact that this specific type of plasmid, PMLST19, carrying genes for resistance to important antibiotics, has been previously found in Salmonella, causing human infections in nearby Uruguay. And Argentina emphasizes the need for ongoing One Health surveillance, looking at humans, animals, and the environment together to track its movement.
It really suggests a potential pathway for resistance to move from poultry into the human population.
It certainly strengthens the argument for that possibility, yes.
Finally, the way the strains clustered based on whether they came from broiler or laying hens, and those geographic patterns within those groups, what's the main message there for, say, control efforts?
Well, it suggests that there might be specific lineages or subtypes of Salmonella Enteritidis that are more adapted to, or perhaps more commonly circulating within particular sectors of the poultry industry, or even within specific geographic regions, maybe even linked to certain companies, given that most broiler samples came from one company. Identifying these kinds of patterns through ongoing genomic surveillance could be really valuable for developing more targeted, more effective strategies to control Salmonella in poultry production. Maybe interventions need to be slightly different for broilers versus layers, for example.
So a really detailed and frankly quite complex picture emerges from this in-depth genomic study. Let's try to summarize the most important things for our listeners to take away from this deep dive.
Okay. In essence, this study of Salmonella Enteritidis from Argentine poultry farms showed us really high levels of resistance to nitrofurantoin, surprisingly persistent, and also concerning levels of reduced susceptibility or resistance to fluoroquinolones.
Right. Two big red flags there.
Definitely. The researchers identified the specific genes and mobile genetic elements, like plasmids carrying virulence or resistance genes, that are associated with these patterns. And perhaps one of the most intriguing findings was that clear genetic clustering of the strains based on whether they came from broilers or laying hens, and also those surprising genetic similarities to Salmonella strains found in human infections in the USA and the UK.
Yeah, that global connection point is really striking.
It really points to potential global pathways of spread.
And ultimately, the value of this kind of detailed research comes down to being able to better understand and hopefully better control the ongoing challenge of antimicrobial resistance in foodborne pathogens like Salmonella Enteritidis, right?
Absolutely. Continuous surveillance, especially using these advanced genomic methods that give us such high resolution, is really critical. It's essential for tracking how resistance is evolving and spreading, both locally and globally. And this information is just fundamental for developing effective strategies to protect both animal health and, of course, public health.
So as we wrap up this deep dive into Salmonella in Argentine poultry, here's a final thought for you, our listeners, to consider. Given the evidence of global interconnectedness revealed by the genetic similarities between the Salmonella strains in Argentina and those found in human infections literally across the world, what broader implications might this study have for how we think about global food safety and public health strategies? Think about the role that international trade and travel inevitably play in the spread of resistant bacteria. What kind of coordinated efforts might be needed, really on a global scale, to effectively address this growing challenge? Something to mull over. Thanks for joining us for this deep dive.